Hey everyone. In this video, we're going to look at some more detail on the process of transcription and RNA processing. Now, if you haven't watched it yet, I recommend you pause here and watch the video on the overview of gene expression before going into this one. The process of transcription happens in three main stages: initiation, elongation, and termination. In initiation, RNA polymerase will bind to a specific DNA sequence called a promoter, which is just upstream of where transcription will start. The details of this process differ somewhat between prokaryotes and eukaryotes. Transcription initiation in eukaryotes requires a lot of additional proteins, including transcription factors, which help to get RNA polymerase bound and to stimulate the process of transcription. Once that has been completed, elongation can start. In this process, RNA polymerase will progress down the DNA, reading the template strand in a 3' to 5' direction and synthesizing a complementary strand of RNA in the 5' to 3' direction. Now, unlike DNA polymerase, RNA polymerase does not need a primer and can start synthesizing RNA from scratch, provided it has a template and free nucleotides. A termination in prokaryotes happens once the terminator sequence has been reached, when RNA polymerase will release the messenger, or mRNA, and the process is completed. Now, since there are no additional steps to be completed and there is no physical separation between transcription and translation in prokaryotes, the two processes can actually happen simultaneously, with ribosomes already translating the mRNA while it's still being transcribed. In eukaryotes, termination happens after the polyadenylation signal sequence has been transcribed, and the transcript is then ready for the additional processing steps that have to happen before eukaryotic mRNA is ready for nuclear export. We're going to look at three modifications that typically happen with eukaryotic pre-mRNA, one at each end of the transcript and one in the middle. First, at the 5' end, the transcript receives a cap, shown here in green, which is actually a modified G residue. The second is at the 3' end, and that's the poly A tail. This is just a fancy term for a long string of A's, usually around 250 nucleotides long in mammalian cells. Together, these modifications serve three main functions. They assist with nuclear export of the mRNA, assist with ribosome binding, and the poly A tail in particular has a protective function, serving as a buffer between the coding sequence and the end of the molecule, so that nucleases, which can digest the mRNA from the 3' end, don't shorten the coding sequence. You've probably noticed that we have two different colors within our mRNA here, and these are the middle sections that will be modified. In its initial form, the mRNA contains both coding sequences, which will be expressed, and so are called exons, and intervening sequences, or introns, which will need to be removed. Let's look at how the cell goes about removing those introns. It's going to use a catalytic complex called a spliceosome, which, among proteins and other components, contains catalytic RNA molecules called ribozymes. This is one of the few instances where we have an enzyme that isn't a protein, although these RNA molecules have several of the same properties which allow them to perform this function. For example, they can fold up into the required shape and can recognize a substrate, both of which is done by complementary base pairing, and they have functional groups capable of catalyzing a specific reaction. The ribozyme can recognize the boundary between an intron and an exon, folding the intron and bringing the ends of the exon together. This continues at each intron-exon boundary until all of the introns are removed and the exons are joined into a continuous coding sequences. Now, the introns will often carry out other functions within the cell, such as regulating gene expression, but only the exons will be used for the purposes of encoding proteins. Now, just because a section of mRNA is an exon doesn't mean it will always be used for translation. The human genome is estimated to contain between 20 and 25,000 protein-coding genes, but can actually produce far more proteins than that. And this is accomplished through a process called alternative RNA splicing, in which certain exons can be included or excluded to vary the proteins coded by the transcript. Let's consider this example here. Our initial transcript has 5 exons, but let's assume that we can pick and choose between exons 3 and 4. In this case, we can make 2 different final transcripts, one containing exon 3 and one containing exon 4 instead. Now, individual exons often encode specific domains or functional regions within a protein. And so you can start to see how, say, including exon 4, which maybe encodes an alpha helical domain, could produce a protein with a partly different structure from one including exon 3, which maybe encodes a domain folded into a beta sheet. Now, should these differences in domain be related to functions like targeting to an intracellular location or binding to a specific substrate, you can start to see how just changing a small part of the protein could potentially alter its function. Thanks for watching. Please be sure to check out the video on translation and the one on gene expression if you haven't had a chance to watch it yet.